So last meeting, we defined a simple diffusion. So we define simple diffusion as a passive transport mechanism. Passive transport mechanism because it does not require the expenditure of energy. For the driving force of the simple diffusion, the driving force is the concentration gradient, meaning that a higher concentration to a lower concentration. So there is a downhill movement of the molecules from an area of higher to lower concentration. So for the simple diffusion, for the molecules that are transported or for the drugs that are transported across the membrane by way of simple diffusion, example of these drugs are the lipid soluble or those drugs that are nonpolar. So when we say lipid soluble drugs, these drugs are able to readily pass across the cell membrane without the aid of transport proteins. So the second mechanism is facilitated diffusion. Facilitated diffusion is a mechanism whereby the drug enters the cell through specialized transmembrane carrier proteins that facilitate the passage of large molecules. So in this type of mechanism, the drug will be able to move from an area of higher concentration to an area of low concentration. So just like the simple diffusion, facilitated diffusion, the main driving force is also the concentration gradient. So this type of diffusion does not require energy. It is also a passive transport, and the carriers may be inhibited by compounds that compete for the same carriers. So we have here a figure showing the plasma membrane. So within the plasma membrane, you can see here the presence of transmembrane proteins. So these are integral proteins that are present within or it is present now in the plasma membrane. So this is the outside of the cell. So we have here the outside of the cell and the inside of the cell. So you can see here that the concentration of the molecule is greater in the outside relative to the inside. So for those molecules that are unable to pass or unable to readily pass across the cell membrane, they will be able to traverse the membrane by way of transport proteins. So in this case, we have here a protein that will serve as a channel whereby these molecules will be able to pass through the membrane from the outside to the inside. So this transmembrane protein carrier changes the shape to facilitate entry and exit of some nutrients. An example of this nutrient is fructose. So fructose is a sugar molecule and it is considered to be a polar substance. So when we say polar, that is hydrophilic and it is not lipid soluble. So since fructose is not lipid soluble, it will not be able to pass through the cell membrane, so it needs a carrier protein in order for it to pass through the membrane. So basically, the main driving force for facilitated diffusion is the same for simple diffusion. The concentration gradient, meaning an area of higher to lower concentration. And this is also assisted by a protein which is known as a carrier protein. So polar molecules cannot pass readily through the cell membrane and they diffuse across the membrane through transport proteins. So a transport protein allows certain molecules or ions to diffuse across the membrane. So we have different types of transport proteins. In facilitated diffusion, we have the channel protein, the gated channel protein, and the carrier proteins. So when we say channel protein, this acts like a pore in the membrane. So when we say pore, that is a an outlet, no, para siyang outlet no, in the membrane that lets water molecules or small ions pass through quickly the membrane. So examples of a channel protein are apoporines and ion channel proteins. So when we say apoporines, no, from the word aqua, these are channel proteins that facilitates the the diffusion of water in the membrane. We also have the ion channel proteins. So these are important channel proteins for ions or charged molecules. So an example of facilitated diffusion that uses water channels or aquaporin is osmosis. So when we say osmosis, we define osmosis as the movement or the diffusion of water through a selectively permeable membrane. So water is a polar molecule. So it is not lipid soluble. So it needs channel proteins in order for it to move across the cell membrane. So this type of channel proteins are known as aquaporins. Aquaporins allow water to pass through the cell membrane. So without aquaporins, water would diffuse in and out of the cells very slowly. So in this case, in this diagram, we have here the cell membrane, and the cell membrane contains the water channel aquaporine. So when, for example, there is a higher concentration of water in the ECF, so the water will move across the cell membrane by way of these channels. We also have gated channel proteins. So from the name itself, it is gated, meaning that it would allow a molecule to pass through the membrane in the presence of this channel. The gated channels have a binding site that is specific for a given molecule or ion. A stimulus will cause the gate to open or shut down. So, for example, when a molecule will attach to the binding site of this particular channel, that channel will be simulated and it will open, and this will allow the ions or the molecules to pass through the membrane. So what are examples of the gated channel protein? So examples of these are the sodium gated channels. Of course, this will allow the sodium ions to pass into the cell. We also have another molecule, the glucose molecule, and these are moved across the membrane through gated channels. For ion channels, the ion channels are also important in facilitated diffusion. So basically, the ion channels in the cell membrane will form pores, and channel has to be open. So it is usually open. There are different types of ion channels. We have the voltage gated, ligand gated, and the mechanically gated. So when we say voltage gated, that particular channel is usually opened or closed depending on the voltage or the membrane potential of the cell membrane. So for example, these are usually present in the neuron and the cardiac muscle or the cardiomyocytes. So for example, when the membrane will be depolarized, this will create an action potential, and that action potential will open this particular voltage-gated ion channel. So when this ion channel will open, so this will allow or this will facilitate the entry of the molecules or the ions from an area of higher to lower concentration. So this is, it can be from the outside to the inside or from or the opposite, no, from the outside to the from the inside to the outside, depending on the concentration gradient of the molecule. The next type is the ligand-gated no ion channel. So basically, in this type of ion channel, we have here a site in the ligand in the ion channel that will be stimulated in order for this particular channel not to open. So for example, when an allosteric site of the channel will be stimulated, this will allow the channel to open and it will allow the ions to pass through the membrane. So another type of molecule that is being transported by way of facilitated diffusion is the glucose. So we define glucose as the simplest form or the basic unit or the simplest form of the sugar molecule. So basically, for glucose to be transported across the membrane, they need uh, transporters. And these transporters are known as GLUTs, no, G-L-U-T, GLUTs. So these GLUT transporters are thought to be involved in sodium-independent facilitated diffusion of glucose into the cells. So when we say sodium-independent, this type of membrane transport is not dependent on the presence of the sodium ions. So insulin stimulates glucose transport by promoting the translocation of intracellular vesicles that contain the GLUT4 and GLUT1 glucose transporters to the plasma membrane. So for insulin or for glucose to be able to move, to move from the outside to the inside of the cell, it needs GLUT4 transporters. And the GLUT4 will only be present in the plasma membrane when insulin is also present in the vicinity. So basically, when we say insulin, that is a hormone that is produced by the pancreas. When it is present, it will stimulate the uptake of glucose by our cells. Now, for example, the liver. So in the liver, GLUT4, when glucose are present in higher concentration in the blood, this will facilitate the formation of our glucose transporters or GLUT4 in the presence of insulin. So in this case, the glucose now will be able to move from the outside of the cell towards the liver cells. And this effect is considered to be reversible so that when, for example, insulin will not be present or when glucose level in the blood is lower, the GLUT4 will also be it will also detach from the plasma membrane so that the transport of glucose from the outside to the inside would also not happen. So how glucose enters a cell? So glucose enters the cell by way of our glucose transporter. And this is an example of facilitated diffusion because this type of transport is from higher to lower concentration. So in this case, we have here our glucose transporters that are present in the plasma membrane. So this is the outside of the cell and this is the inside of the cell. So again, for facilitated diffusion to happen, there should be a higher concentration of glucose from the outside to the inside. So for example, when glucose will bind to the, for example, our GLUT4 that is present in the liver, for example, the glucose will cause the protein to change the shape and exposing the glucose to the inside of the cell. So there is a change in the shape of the protein and the glucose will be able to move inside the cell. So glucose will pass into the cell and into the protein, returning to its original shape. So after the glucose enters the cell, so magbabalik ngayon yung shape ng ating transporters. So pag bababa ngayon yung concentration ng glucose sa ating plasma or in the ECF, these transporters will detach now from the plasma membrane so that the glucose here will not be able to be moved towards the inside of the cell. So para hindi maubusan ng glucose sa blood, sa dugo. So carrier protein, another type of protein that will facilitate or that is involved in facilitated diffusion is the carrier protein. So this type of protein will transport ions, molecules, or group of substances. So they carry, from the word itself, carrier, so they will carry the ion or molecule across the membrane by changing the shape after the binding of the ion or molecule. So this particular type of carrier protein, this particular type of transport protein is not only present in facilitated diffusion, but it also acts on active transport. So these are the examples of the different proteins, transport proteins that are involved in facilitated diffusion. So in here, this is an example of a protein channel, and these three here are examples of carrier proteins. So in this diagram, we have here an illustration of our facilitated diffusion, and we can see here that this left figure here, so we have here the presence of our transport proteins. And this type of protein is a channel protein. On the right, we have here another type of transport protein which are known as carrier protein. The difference between the two is that for carrier protein, in order for the molecules to be able to enter the cell, there is the change in the shape of the protein in order to accommodate the shape also of the molecules. So the same, pareho lang sila ng driving force, which is the concentration gradient, so from higher to a lower concentration. So pag halimbawa, the equilibrium, so after equilibrium will be achieved, so this carrier protein will now close after the equilibrium of the molecules has been achieved. So comparing the simple diffusion to facilitated diffusion, so basically they are both a passive transport mechanism. They do not require energy. But for facilitated diffusion, carrier proteins and transport proteins are present. This is not present in simple diffusion. The next type of drug transport mechanism is active transport. So from the word itself, active, meaning it requires energy. It is energy dependent and it uses ATP in order to achieve its function. It also involves the presence of specific carrier proteins. So it is capable of moving drugs against a concentration gradient. So that is an uphill movement of a drug molecule, meaning from an area of lower drug concentration to a higher drug concentration. So an example of this, in this figure, we have here an active transport mechanism. So an example of those molecules that are being transported by way of active transport are, we have the minerals, sugars, and amino acids. So they move against a concentration gradient, meaning from an area of lower to higher concentration. and this requires the presence of ATP. So this is a type of a carrier protein. So ano ba yung ating example ng active transport? Active transport mechanism. So an example of our active transport mechanism is the sodium-potassium pump. What is the function of the sodium-potassium pump? The body? Any volunteer? Ano yung function ng ating sodium-potassium pump? So to transport sodium and potassium ions across the cell membrane. So specific, ano yung specific niya na function? So where does sodium go and where does potassium go? Saan ba pupunta yung yung ating sodium? Saan pupunta yung ating potassium? So normally, during rest, at resting membrane potential, ano gani yung voltage ng cell at resting membrane potential, particularly in the neuron? Ano yung voltage ng cell sa resting membrane potential? So normally at resting membrane potential, the voltage of the cell is negative 70. So this means that there are more positive, the outside of the cell is more positive relative to the inside. So yung inside ng cell is negative. The sodium ions are normally concentrated. Saan sila mas concentrated yung ating sodium ions? Outside or inside the cell? So usually yung ating sodium ions, they are the major cations in the extracellular fluid. So they are more concentrated in the outside of the cell, about 150 milliequivalent or millimoles. They are present there at higher concentration. So they are the major cation in the ECF, while the potassium is the major cation in the inside the cell or in the ICF. So what the sodium potassium pump will do is that it will pump 3 sodium ions in exchange for 2 potassium ions. So this pumping of sodium outside of the cell and pumping of in exchange for 3 sodium, there will be an entry of 2 potassium ions. This is important to maintain the resting membrane potential, wherein the outside of the cell is relatively positive, is positive compared to the inside, which is negative. So that is an example of an active transport because it uses energy in order for it to be, in order for it not to materialize. And the movement of the molecule is also from an area of lower to higher concentration because when you are going to move sodium from the inside to the outside, of course, you are against the concentration gradient because sodium is usually concentrated in the ECF or the outside of the cell. When you are going to move potassium from the outside to the inside, so of course that is also against the concentration gradient because potassium is normally more concentrated on the inside relative to the outside. So ano siya? Opposite the concentration gradient. So that is an active transport. Another type of active transport is these are classified as bulk, BULK, bulk transport mechanism. This type of, these are the endocytosis and the exocytosis. So this type of drug delivery transport transports drugs of extra large size across the cell membrane. So in endocytosis, this involves the engulfment of a drug molecule and transport into the cell by squeezing the drug-filled vesicle. So in this figure, we have here a vesicle that is filled with a drug, and the vesicle can bind to the cell membrane. It will be engulfed into the cell to form a vesicle or an endosome. For the exocytosis, that is the opposite of our endocytosis, and this is used by the cells to secrete many substances by a similar vesicle formation process. So we have here a secretory vesicle inside the cell, and this secretory vesicle will move towards the plasma membrane, and it will unload its contents towards the outside of the cell. So that is an example of an exocytosis. So certain neurotransmitters are stored in the membrane-bound vesicles in the nerve terminal and are released by exocytosis. So an example of a neurotransmitter is the norepinephrine. So usually, norepinephrine are made, they are present in the, they are usually present in the nerve terminals of the noradrenergic neuron. So the other name of norepinephrine is noradrenergic. So within this neuron, there are vesicles that are present in the inside at the nerve terminal of the neuron. So these vesicles contain the norepinephrine. So when this particular neuron will be stimulated, this will stimulate or facilitate the release of these vesicles, unloading its contents to the outside of the cell, unloading the neurotransmitter norepinephrine to the synaptic cleft. And this norepinephrine, after it's released to the synaptic cleft, it can bind to the receptors in the target organs. So that mechanism is can be classified or as an example of exocytosis. So these are the two major groups of transport processes across the cell membrane. We have the passive and the active transport. For the difference between the two, for passive transport, substances are moved from higher to lower concentration. For active transport, the substances are moved from lower to higher concentration. That is an uphill movement. For the passive, this is a downhill movement of molecules. So this passive transport will not require energy. Active transport will require energy. There are three examples of passive transport. We have the simple diffusion. Simple diffusion is true for lipid-soluble molecules. Facilitated diffusion is can be applied for example, the ions, no, for and other polar molecules. Osmosis is for water. Active transport examples are protein-assisted or carrier-mediated protein transport. For example, we have the sodium-potassium pump, endocytosis, and exocytosis. So before we proceed to the next topic, we'll have first a video presentation on the different membrane transport mechanisms.
All animal cells are enclosed in a plasma membrane, which consists of two layers of phospholipids. The hydrophobic nature of the cell membrane makes it intrinsically permeable to small,